Hello viewers, welcome to this biology revision series. Today we are going to deal with the cell biology. So basically this is a question that came in 2025 exams, that's paper 2 biology. So let's see what the question is requiring us to do. Question 1 says, Figure 1 shows a PALISADE cell observed under a light microscope. So now, one thing that you should be very much careful about is read the statement clearly. So figure 1 shows a PALISADE cell observed under a light microscope, and the figure has been displayed here with labeled letters A, B, C, and other structures like the cell membrane, the nucleus, the cytoplasm. So now, question A is requiring to identify the parts labeled A, B, and C in figure 1. So the first thing you should keep in mind is that this is a PALISADE cell and it has been observed under a light microscope. So you should be able to know the difference. We have two types of microscope. We have an electron microscope and the light microscope. The light microscope just helps in the visualization of the entire cell, and it doesn't have better resolution power, meaning it doesn't really have an ability to differentiate close objects. Okay, so it's more like giving us the general structure of the PALISADE cell as demonstrated there. So we want to identify the parts labeled A, B, and C. So let's try just to zoom in and label these structures well. So we have A, B, and C. What are the structures labeled A, B, and C? So the structure A, we can clearly see that A is the cell wall. Okay? Then how about B? B is pointing at the vacuum. And then how about C? C is pointing at the chloroplast. Okay? C is pointing at chloroplast. So C is pointing at the chloroplast. We have identified the parts that we were supposed to identify. Then let's go to another question B, which is saying, explain the functions of part labeled A. Okay. Explain the functions of part labeled A. You should be able to look at mark allocation just one. So you need to give a brief explanation. One of the functions of part labeled A is the cell wall. It provides strength and support to the cell. And then the other function of part labeled A is that it provides the cell shape, or rather it maintains the shape of the cell. So these are basically two functions of the cell wall, but here it's only required you to write one function of the cell wall, and then you get that 1 mark of yours. Okay. Then how about question C? Question C is saying, give two differences between the cell in figure 1 and that one found in the human tract. So the cell in figure 1 is a specialized plant cell, the PALISADE cell, and then that found in the human trachea is also another specialized animal cell. So this question is more like trying to state some differences between the PALISADE cell and then the trachea cell. The cells that are found in the trachea, probably we have the ciliated cells that traps dust. So what differentiates that ciliated cell from this PALISADE cell? One difference is that the cell in figure 1 has what we call the cell wall. The cell wall. Wow, that is found in the trachea does not have. Then what is the second difference? The second difference is that the cell in figure 1, the cell in figure 1 has what we call large vacuum. Okay, so this one has large vacuum, while that cell that found in the trachea doesn't have a vacuum. Okay, so these are the noted two differences that you can state between the cell in figure 1 here, the PALISADE cell, and that of a trachea. Then I'm going to give you the bonus, the third difference. The third difference is that this cell in figure 1 contains chloroplast, the one we're just labeling, chloroplast. Wow, because this chloroplast is very much important in the process of photosynthesis. However, that cell that found in the trachea do not have. So I've given you three differences. You are going to choose for yourself. Then now, let's look at D, which is saying, state the names of any other two organelles that would be seen if the cell in figure 1 was observed under an electron microscope. So initially, if you followed through, this structure was viewed under a light microscope. So you should be able to say the differences between an electron microscope and the light microscope. So I stated to say the light microscope just have the viewing power, like be able to visualize the entire cell, and if you want to view the cell in colors, it gives that ability. However, with an electron microscope, you can only view the cell black and white, basically. And then one notable difference that is going to help us answer question D is that light microscope has lower magnification, okay, about 1500 to 200 times X, okay. However, with the electron microscope has about 2 million magnification, 2 million times. It can view a particular structure 2 million times. That is bigger than the structure that is normal. That's what we mean by magnification. So when you look at these organelles that have been presented here, these ones have been observed under a light microscope. So there are some structures which can't be differentiated well. That's why we're able to see just the chloroplast, the nucleus, and the cell membrane. So this particular cell have the electron has the ability to view some organelles that are smaller or minute to be seen under the light microscope. And amongst these organelles, we have Golgi bodies, okay, or Golgi apparatus. And then apart from Golgi apparatus, we also have mitochondria. Mitochondria can also be viewed under light microscope. Apart from mitochondria, we have smooth endoplasmic reticulum. Smooth endoplasmic reticulum, they can also be viewed under electron microscope. Apart from that, we also have ribosomes. The ribosomes can also be viewed under the electron microscope. But this question required only to give 2 organelles. So you can choose amongst these 4 that I've given to you. Alright, thank you very much for following through. Don't forget to subscribe so that you don't miss content, other videos that I'm going to post in other courses and in this course. Bye bye.